The Distinguished Alumni Award is the highest award given by the School of Public Health. It recognizes alumni with a demonstrated record of distinguished service and achievement in a public health discipline or setting. And tonight we honor Dr. Bruce Psaty with the 2018 Distinguished Alumni Award. You'll hear from Bruce on tales of drug safety and advances in regulatory science at the U.S. Food and Drug Administration. And afterwards, we'll have a period of question and answer that'll be moderated by the chair of epidemiology, Steve Hawes. So I'm going to ask that you hold your questions until we have that moderated Q&A period at the end. Staff will also be available to circulate to collect your questions towards the end of the lecture, so that if we want to have Steve address them directly, that we can do that as well. Bruce Psaty earned his MPH from our Department of Epidemiology in 1986. This was not the first degree that Bruce earned. He earned his undergraduate degree from Princeton University and then received his MD and his PhD and his master's in the arts degrees from Indiana University after that. Throughout his career, Bruce has made exceptional contributions to the school's mission of research, teaching, and service, beginning with the development of the school's first course in cardiovascular disease epidemiology in 1987. He's a national leader in encouraging better post-market surveillance of approved medications, and he has advocated for advances in regulatory science at the U.S. Food and Drug Administration. We'll hear more about this in his talk tonight. As many of you know, he's a professor in departments of medicine and epidemiology and in health services. He co-directs the University of Washington's Cardiovascular Health Research Unit. In addition to the award tonight, which of course we hold especially dear, he's been awarded many awards by many associations and organizations throughout the course of his career. In 2004, he was the Remington Methodology Lecturer at the AHA Epidemiology and Prevention Council. He received our school's award for Outstanding Faculty Community Service. 2005, he achieved the University of Washington-wide Outstanding Public Service Award for his work in drug safety in the same year. He was the Ansel Keys Memorial Lecturer at the American Heart Association Scientific Sessions in 2009, served as a member of the U.S. Food and Drug Administration Science Board in 2011, and as a member of the NHLBI's Advisory Council from 2012 on. In 2013, he received the highest award that the American Heart Association gives to a scientist, the Distinguished Scientist Award. And in 2013, he was also elected to the Institute of Medicine, now the National Academy of Medicine. He's been an investigator in a variety of fields in addition to the drug safety we're going to hear about tonight. He's published more than 850 articles, editorials, and commentaries, and recently he has led a huge collaborative effort with investigators from a variety of cohort studies to establish the CHARGE Consortium, the Cohorts for Heart and Aging Research in Genomic Epidemiology. So often when you look at a CV like his, not only are there 850 articles, but it seems like some of the papers have 850 authors on them. And you wonder, what did that person do as part of authorship of that paper? In Bruce's case, in most occasions, the work would not have happened without his organizing that consortium and making it happen. So it's very clear with the work that he's done. His work has led to new genetic associations and potential targets for new drug therapies. And we could go on and on and on. But you're not really here to hear from me, you're here to hear from Bruce. So I want to congratulate Bruce and thank you for your contributions to the health of our communities. And Bruce, take it away.
I think it's on. Thank you very much for that lovely introduction. I had somebody wrote me earlier today and said, boy, you haven't aged in 10 years. And one of the secrets to that is using a picture that's 10 years old. So can we switch to the slides? Very good. selection as the distinguished alumnus is a high honor, and I'm very grateful to the school and all the collaboration and mentorship I've received here. So I think of it as a celebration of the School of Public Health and epidemiology and health services as well, and even biostatistics. So you might wonder why I chose this. Well, the dean's office told me what to talk about today. And in their wisdom, that means you don't have to hear about things like case control studies, large-scale collaborations, population biology, precision medicine. So they've spared you a lot of discussion. So in terms of work with pharmaceutical industry, I'm almost entirely clean and sober. There are some minor exceptions that I can discuss later if you want to hear about them, but I would call myself clean and sober. I've served on the FDA Science Board now since, I guess, 2011. I thought it was since 2012. They keep upping my my tenure there. And it's really a terrific organization, the FDA. They have an unimaginably difficult task, regulating not only drugs, but devices, vaccines, biologics, food, and tobacco. And there are a tremendous number of hardworking people there, and we owe them a lot. The goals of this talk are really three. One, to honor mentors and collaborators. And I'm going to tell you some drug safety stories, most of which I've been involved in, and then describe some of the evolution at the FDA. There are several themes I'll touch on. One is the tension between the speed of approval of a drug and the quality of the risk-benefit information. The approval is a major step, and once a drug is approved, companies can market it off-label for any indication they want. There's very little ability to restrict that. So the approval marks a major transition. And if the information isn't obtained prior to approval, it's often difficult to obtain after approval. So fast drug approval may mean it may get a drug to the public more quickly, and some people may need it. It may be life-saving for some people, but the risk-benefit information would be compromised. The other, this may seem obvious, but I'll try to illustrate it. Unexpected safety problems are invisible, and it's very easy for hundreds of thousands of people to be injured without it's being noticed, if there isn't some sort of surveillance system or somebody looking. And finally, the public health value of science-based regulation, and I think the FDA does a terrific job. I quibble with them now and then on certain things that others do, but for the most part, they do terrific work. So as I said, this is to honor mentors, and there are two here. Jim, does this work? Oops. This may be beyond my technical capabilities. The laser doesn't seem to be working. Jim Legere, this is the Robert Wood Johnson Clinical Scholars class of 84, 85. Jim Legerfo and Tom Inouye were mentors at that time, and I still work with Jim in the adult medicine clinic. I would point out that three other people in this picture are former award winners of the Distinguished Alumnus Award: Michael Phillips, Mary Bassett, and Art Kellerman. So if anybody has a pointer, a laser pointer, they might bring it down. I do need it later. So we're in an academic institution, so I thought I'd start with a quiz and invite you to think about the early 21st century and what can the FDA do legally? Can they withdraw a drug, suspend sales, compel the completion of post-market study, control advertising, revise the product label of a drug? What sort of authorities did they have in the early 20th century? 1, 10, all 5? None. FDA could do none of these things. Yeah. Biostatistics comes through. Where was Epi? So just a brief history. Drug disasters tend to lead to federal acts. So sulfa drug was laced with basically a poison. It led to the Food, Drug, and Cosmetic Act in 1938. Drugs had to be tested for safety at that point. And then the Kefauver-Harris amendments were precipitated by the thalidomide crisis. This is a picture of Frances Kelsey receiving an award from President Kennedy. She was so slow in approving this drug that it prevented its coming on the market in the US and spared tens of thousands of patients. Terrible babies, terrible birth defects. I myself was not able to put examples of that on this slide for you. So the FDA approval process, the goal is to determine that a drug is safe and effective for its intended use. And there are a series of steps. It includes phase 1 trials, which are in normal people. Phase 2 studies, which are safety studies in patients who have the condition under study. And then phase 3 studies, and they often don't have up to 3,000 people. She's sweet. Those are the major studies, randomized trials to show the drug is safe and effective. The FDA review and then the approval occurs. And there are often or sometimes phase 4 studies that are post-market commitments. Sometimes they're done, sometimes they're not done, sometimes they're incomplete, sometimes they get turned into marketing studies of various sorts, and the control over that post-market process is partial and difficult. So how did we approve drugs in the late 20th century? Well, we did a high quality, in the US, high quality pre-market study, what I showed you before with the phase 1, phase 2, and phase 3 trials. And at that time, approval marked a transition. There was basically evaluation, approval, and then marketing. And that's the, boo. Studies, so approval marked the transition from evaluation to marketing. We conducted things much more leisurely than they did in Europe, and this was quite good, because under this model, if a drug could get approved in Europe, come on the market, be shown to be toxic, and be taken off the market in Europe even before it was approved in the US. And Practilol is an example of one of those, and a number of drugs just never made it on the US market because our approval process was so thorough and so slow, they didn't make it. So I really admire the work that the people at the FDA do. This is John Sr., who's a hepatologist, who's been at the FDA for years and studied drug-induced liver injury. And he wrote us when we were working on the IOM report, The Future of Drug Safety. And this is what he described happens at the drug companies. And I think it's very important. Most companies assemble teams of scientists to develop new drug products and win approval, but disband and reassign the team members after approval and turn responsibility for the product over to the sales and marketing staff, who have little medical expertise or concern for safety problems that they perceive as simply impediments to the marketing process. So this is the culture at pharmaceutical industries. So what happened in the US? Well, we had HIV and cancer activists in the 1980s said we need to bring drugs to market more quickly. So Congress came up with the Prescription Drug Fee Users Act. First one was in 1992. And between 1992 and 2001, the review times for drugs dropped from 33 months down to 14 months. So that's more than half. So they sped up the approvals considerably. And drugs came onto the market first in the US, new chemical entities, at a rate of about 2% in 1980. By 1998, 60% of new drugs were coming on the market first in the US. So America became a drug safety testing ground without a robust safety system. We actually relied on the integrity of industry for safety, which you've seen John Sr. describe as an impediment to sales. So I think you can see where this might be leading. As an additional note, the Prescription Drug Fee Users Act prohibited the use of user fees for drug safety. Over the next 10 years, the Office of Drug Safety at the FDA shrank because its budget that wasn't user fees had to go elsewhere. So by 2000, 2002, the Drug Safety Office was as small as it had ever been. So I'm going to go through 3 drug safety stories tonight. One is Cerivastatin, Baycol. It's a lipid-lowering drug. It's one of the statin drugs that I'm guessing 50% of the audience might be on. And it is associated with, just based on age, it's associated with muscle injury. I'm going to talk about surrogate endpoints. This is not nearly as exciting because it's a complicated concept, but it's so much more important from the point of view of public health and whether drugs work well for the population as a whole. Then I'm going to talk about rofecoxib, Vioxx, a nonsteroidal anti-inflammatory drug in heart attacks. And this is the drug safety event that kind of led to the FDA Amendments Act and yet another cycle in drug safety problems followed by revisions in the drug laws. So again, I want to honor mentors. Tom Kepsel and Ed Wagner have worked with me from the beginning on many of these studies and guided me. Early on, I did a single study at Group Health. And that single study had 2 papers. One suggested that beta blockers might be better than high-dose diuretics for the treatment of high blood pressure in terms of the risk of heart attacks. And the other suggested that stopping beta blockers stopping them abruptly was associated with an increased risk. And I was struck and unprepared. So the EPI courses don't tell you how to deal with the media when they come to chat with you about the study. And the media, of course, wanted the drug safety story. And they don't tell you how to deal with industry when industry comes to you and wants you to help promote their products. And I was struck by these were published quite close together. I was struck by the difference. This is one study, and we're talking about putting together risk-benefit profiles of a drug. And these are both part of the story, but they weren't interesting to the groups, to both groups, and the interpretations perhaps lack some nuance. So Cerivastatin is a cholesterol-lowering statin drug. It causes what's called rhabdomyolysis. It's a breakdown of muscle cells that can cause kidney failure and death. The injury to patients went unrecognized by the FDA, but not by the manufacturer. At this time, rhabdomyolysis was recognized as a side effect of the class of drugs called statins. And so the FDA didn't even look for it. They didn't pay any attention to it. The only thing they could do was look for new drug side effects. How did I know that the manufacturer knew about this? Well, I used a novel source of data. I was an expert witness for plaintiffs suing Bayer for rhabdomyolysis. And I reviewed the records that were available to the lawyers, and it was clear to me that within 6 weeks of the drugs coming on the market, this drug was incredibly dangerous and was causing harm. Within 6 weeks, this was obvious. But I was stuck. The company's behavior was completely inappropriate. The drug interaction with gemfibrozol, another lipid-lowering drug for triglycerides, was absolutely clear. And they did not put that on the label for about 1.5 years. They waited at least 1.5 years. And the behavior was completely inappropriate. I had signed a non-disclosure agreement, which means that if I reveal any of this, they can come after me. So I was reading the New York Times, and I was reading about a lawsuit in Texas. And it turns out it was about Cerivastatin. So I called up the Nueces County clerk and asked if there were public documents, the what do they call them? The exhibits, yeah. And yes, they were available, and it would cost me $1 a page. So I wrote a check, or Sue did, for $400 or $500, and I got all the exhibits. And with several other people, including Noel, we wrote an article describing what the company knew and when they knew it. And I think one of the most important features of this work is the protection provided by the university. So I was at risk for having high-paid lawyers come after me. My grants don't include lines for, lawyers. And Pat Wall at the time, who was dean, Bill Bremner, who was chair of medicine, Steve Finn, who's here tonight, and Kathy DeAngelis, all helped to protect this publication and to allow me to move it forward. That was very important. So this slide is from one of the exhibits. In patients who are on gemfibrozol, Cerivastatin compared with atorvastatin was associated with an 855-fold increase in the risk of this serious adverse event. Now, I've been reading medical articles for 35 years, and I think I've only seen one that's higher, ever. This is an incredible risk. And did the company understand this? Well, this is their table, basically. So this is what the epidemiologist from Bayer said. Findings indicate that in patients receiving monotherapy, Cerivastatin substantially elevates the risk for rhabdomyolysis compared with other agents. That's fair, 20, 30-fold. In combination with gemfibrozol, Cerivastatin patients were also found to be at remarkable disadvantage compared to patients receiving gemfibrozol with another statin. So it's absolutely clear they knew what was going on, and they failed to report it in a timely fashion. After this paper was published, I got to know some of the lawyers. I don't know that I ever met them, but I corresponded with them. I got to know a number of the lawyers in the UW office, and they would review all drafts of the paper that went in and so forth. And one of them sent me the link to this Bayer security litigation. So Bayer was being sued by the New York Retirement Association because their stock had plummeted. And this is a description of what happened within the company. This is on August 2nd, 2000. A consensus had emerged. This is several months after that slide I just showed you. The data concerning Bacol's dangers was putting the brand at risk. When that conclusion was communicated to Ebsworth, he dismissed the reservations of the safety experts and instructed his marketing team to promote the hell out of this product. So, what do you make of this? It's at worst a purely economic decision. It's also possible, I have several other explanations. One other, maybe kinder explanation is that somebody told them that observational studies may not always be reliable and you can't trust them. You need randomized clinical trials. And for many things you do. A relative risk of 855 is not one of them. But, for many things you do need them. So he might have been persuaded by that, because in discussions with actually some of the executives at Merck about Vioxx, what I discovered is these drugs are kind of like their children, or maybe their grandchildren. You have to be very careful. If you call their drug ugly, troublesome, and a menace, they get very upset. I mean, they just don't want to believe it. It's just so that this is moving the company forward. And so my conclusion for this is maybe a little too soft. Industry may lack a symmetric interest in efficacy and safety. Say again? I don't want to be unfair. So I want to talk about surrogate endpoints briefly for evaluating risk factors such as high blood pressure, cholesterol, and glucose. So this figure illustrates the association that we see in an observational study between the level of a risk factor such as blood pressure and the rate of, say, heart attack events here. And so the higher the blood pressure, the higher the rate. And the idea for blood pressure lowering is that if you lower the blood pressure, you lower the risk. That's the underlying idea. Now, if you can conduct the trial solely with the outcome of blood pressure, you can do it in a couple hundred patients in 6 or 8 weeks. If you actually have to prove that your drug reduces the rate of clinical events, you need thousands of patients for 5 years, say approximately. So it's a much more complicated and difficult approval process. So companies like to use the surrogate endpoints and just assume that you will benefit. Some of us prefer to have evidence that the drugs actually do what they're supposed to. And here's an example of that kind of problem that can occur. These are the phase 3 trials, the approval trials for rosiglitazone. Rosiglitazone is a diabetes drug, and so you can see that compared with placebo, it reduces fasting glucose. That's what a diabetes drug should do. It reduces hemoglobin A1C, but it increases the bad cholesterol and increases weight significantly. So what's this going to do to heart attack risk? Or actually, what it'll do to the pharmaceutical industry is result in more drug sales because you have to treat the weight change and the LDL cholesterol. But it's not clear that this is a favorable risk-benefit profile. And I'll come back to this drug later. So medical care can be a little bit mysterious. This is a slide representing time trends in antihypertensive drug treatments. There are 4 drugs represented here: diuretics, beta blockers, and then 2 newer classes of drugs, calcium channel blockers and ACE inhibitors. And what happened here is that a trial was published, the SHEP trial, in 1991, showing that low-dose diuretics prevented not only heart attacks, but heart failure and stroke in older adults. And what do you see? The use of this drug drops precipitously from 60% down to 40%. Now, how does that happen? Is that miraculous? So the way it happens is there's a kind of triumvirate in each area. And the triumvirate consists of the pharmaceutical industry, marquee professors paid to promote the drug, and then the professional societies. And we have these in lipids, in hypertension, in opioids, and a whole lot depends on the drug. So for the statins, in general, the statins are a good drug, and so when the lipid mafia is promoting that and more people are taking it, that's probably advancing the health of the public. In the case of opioids, it's been a total disaster, right? We've got tremendous use of that, overuse of those. In the case of high blood pressure drugs, these companies said, look, lowering blood pressure works. It really works. So use our drug, which is actually more expensive than diuretics by several orders of magnitude. And they were able to persuade physicians to do that. And so these were all approved on surrogate endpoints, just blood pressure lowering. There were no event trials. And so one of the things we did is we asked the question about whether the use of whether observational studies could help us answer that question. So the ideal thing would be to have a randomized trial showing the health outcomes for heart attack. We had the surrogate endpoint trials, and what we tried to do is get endpoint information, but use observational studies. And collaborators here included Noel, Kurt Furberg, Susan Heckbert, and Fritz Rosendahl from Leiden. And our studies suggested that the short-acting calcium channel blockers were associated with a higher risk of cardiovascular events than the low-dose diuretics. You know, it's not a perfect study, but there was no evidence about events for the calcium channel blockers or the ACE inhibitors at this time. This turned out not to be a popular finding. And so I'll use any occasion to write about things. So we wrote about the attacks. Ed Wagner was instrumental here in helping out. And, you know, I think we did some good. Warnings were added to the product label for short-acting nifedipine. We really helped draw attention to the value of large long-term clinical trials rather than the short blood pressure trials that the companies were getting away with. Subsequently, I worked with Thomas Lumley and others. Ken Rice was part of this as well, looking at a meta-analysis. It was a network meta-analysis, the first application of this new technique to evaluate the outcomes in clinical trials. So this is a meta-analysis of randomized clinical trials. And indeed, the low-dose diuretics tend to look better here compared with the calcium channel blockers. So we were perhaps not far off in our observational study. So this is a nice piece from Harlan Krumholtz and a review of surrogate endpoint trials. About half of all drugs approved now are approved on the basis of surrogate endpoints. Half. This is a review. 27% of the surrogate endpoint trials that had occurred had a subsequent outcome trial. So you could actually compare whether there was a match between the surrogate endpoint, like blood pressure, and the events endpoint. Only less than half, 46%, actually had a concordant result between the surrogate endpoint and the cardiovascular endpoint. And so surrogate endpoints may not be reliable measures of drug efficacy, and maybe these surrogate endpoint trials ought to be used to rule out a drug that should move forward and identify drugs where clinical trials are then needed. So the third story is rofecoxib, Vioxx. It's a nonsteroidal anti-inflammatory drug. So I always wanted to skate with the grace of our Olympic champion. This drug was approved in 1999, withdrawn in 2004. Sales were as high as $3 billion a year. And it was heavily marketed, heavily advertised, and actually did quite well. This is a review of the new drug application for rofecoxib. Villalba was the medical officer. And even before the drug was approved, it was known that there might be a cardiovascular risk with this drug. She says in 6-week studies, there was 1 event in the placebo group, 0.24%, and a total of 12 events, 0.67%, in the rofecoxib group. Now, this is a very small sample size, but you're looking at a risk that's about 2.5 times higher in the drug treatment group compared to the placebo group. And she says correctly, with the available data, it's impossible to answer with complete certainty whether the risk of cardiovascular thrombotic events is increased. Now, what one would like to see happen is that you actually get a trial to evaluate that. That would have been desirable. And it turned out it was never done. There was no trial to evaluate the risks and benefits. Now, how did they get caught on this one? Well, they actually did trials to evaluate other outcomes, the prevention of colon polyps, and there they needed to use a placebo, and also studies to prevent cognitive decline. So they've got the drug approved for arthritis and pain, and they're trying to get other indications and expand the indications. So they need to do placebo-controlled trials for this. I'm just going to focus on the cognitive function trials. This is work I did with Dick Cronwall. And Dick was actually a plaintiff's attorney for rofecoxib. I was not. I was clean on this one. And Dick passed to me exhibits that were public. And I used those exhibits in consultation with him, we were co-authors on this, to write about what Merck knew and when they knew it in terms of the Alzheimer's trials. So remember, this drug was withdrawn in September, I'm sure you know, September 2004. This is April of 2001. The intention to treat analyses for Alzheimer's, there were 2 of them. In one, the increase in the risk of mortality was 4-fold, 4.4-fold. In the other, it was 2.6-fold higher risk of mortality. If you combine them, it's, in a meta-analysis, it's a 3-fold increase in risk of total mortality. How does this happen? The FDA was on it, and they tried very hard. And so there was back and forth with the sponsor. The FDA review and the sponsor's reply, FDA says, have the ethics committee commented on the ethics of continuing the Alzheimer's trials in view of these findings? And Merck replies, the mortality findings are small numeric differences. There is no data safety monitoring board, which is totally ridiculous. Merck Research Labs has not provided these data to the individual IRBs because Merck does not believe that a safety issue has been identified. So again, I don't want to overstate this, but a studied inattention to potential safety issues may delay their identification and harm patients. So the response to the rofecoxib disaster, it was pulled from the market in September of 2004. By November, Senator Grassley held a finance committee hearing. And I was asked to be part of that, and I was pleased to be part of it. I'm not quite sure how I got there. I was interviewed for about 45 minutes by his staff, and they did not want to know whether I knew what a relative risk was, or a meta-analysis, or an odds ratio. They wanted to know everything about potential conflicts of interest. So those earlier drug encounters with the drug companies led me to be largely clean and sober, and so I qualified. I didn't need to know much, but I qualified. The real star was David Graham. He did a terrific job, and this is part of his testimony. He estimated that there were about 100,000 extra heart attacks due to rofecoxib. And let's see if I can see this with my bifocals. He said it would be the rough equivalent of 500 to 900 aircraft dropping from the sky. This translates to 2 to 4 aircraft every week, week in and week out for the past 5 years. And this adverse event went unnoticed for a long time. The hearing evolved in the long run into an IOM committee. The FDA convened an IOM committee, and I was pleased to be part of that. The IOM review thought the pre-approval activity was limited but high quality, but the post-approval efforts looked largely like marketing efforts. Agreed upon studies were often not completed, so the sponsor would agree to do a post-market study and it wouldn't get completed, and the FDA had no authority to make them do it. Completed studies were designed for marketing. If Joel had given me more time, I could give you good illustrations of these, but he didn't. They exaggerated the reporting of benefits, and there was rapid uptake of some of these with direct-to-consumer advertising. The IOM committee recommended a life cycle approach, an ongoing evaluation of efficacy and safety during the entire market life of the drug, systematic approach to identifying and verifying safety signals so things wouldn't just be skipped over, and kind of the active pursuit and management of knowledge about risks and benefits. This resulted in the FDA Amendments Act, and I was pleased to be part of the release team for the IOM committee. We briefed 4 or 5 committees, and I worked with several staff in the House and the Senate reviewing drafts, multiple drafts of the Amendments Act as it went through. And it provided new authorities to require post-marketing studies when safety problems occurred. They could make label changes. They could restrict the distribution and use of drugs. They had civil penalties of up to $250,000 for violations, and a mandate to establish an active post-market system. Soon after this report came out, Margaret Hamburg became the FDA commissioner. She was there from about 2009 to 2015. She embraced this. The FDA began publishing rationales for their decisions. They developed the Sentinel Initiative, and they developed an interest in what was called regulatory science, thinking about how basically using their experience and their science, and they use science-based reviewers in many cases. If you think of some of the things they need to review and approve now with the new biologics and the new genetics and CRISPR and so forth, the FDA reviewers need to know about this science. So they use active scientific folks to participate in the review and develop and change in the approval criteria. So I want to go back and just to the surrogate endpoint part. This is the ACCORD trial is a trial of intensive glucose lowering. It used rosiglitazone, the drug I talked about earlier that decreased the glucose but increased LDL cholesterol and weight. Well, guess what? It failed to prevent coronary events. It increased weight and low glucose episodes so people would pass out. And it actually increased total mortality by 22%. So in part as a result of this, the FDA released a new guidance on the approval of drugs for type 2 diabetes. And it was complicated, but basically it required some evidence about the level of cardiovascular events and the risk of cardiovascular events, and not just whether it lowered glucose or hemoglobin A1C. So this is my last slide. So to go back to the themes, speed of approval versus quality of information. We need to continue to have clinical trials, largely of endpoint trials. I think they need these in cancer too. They're doing the new basket studies. And we need to know how these drugs work in some of the specific tumors. And they need to continue post-market surveillance. The invisibility of unexpected safety problems. So this is really to emphasize the importance of epidemiology and publicly funded public health scientists who do this kind of work. And there are other people here at the University of Washington who do it in pharmacy and health services, epidemiology, at group health, locally. I'm sure there's some at the Hutch. And people across the country, Wayne Ray in Tennessee, Jerry Avorn in Boston, Aaron Kesselheim. There are a number of people who do this. Many of them work with the FDA on FDA committees, but they also keep their eye on the FDA and write about what's happening and whether what's happening makes good public health sense. And the value of science-based regulation at the FDA. This helps redirect some of industry's less noble incentives. And I think it leads to a better risk profile for new drugs. So I'd like to end by thanking other collaborators and family, Nana, Nick, Lou Culler, Sue, Rose and David, James Floyd, Josh, Peter Reitzma, Sarah, Jen Brody, Mandy, Eric Larson, Ken Rice, Russ Tracy, Emily, Barbara, and Robert. So I'd be happy to take any questions. Thanks.
Thank you so much, Bruce, for that wonderful story about your experience. And as I believe we'll start gathering questions from the audience. If you have those, please pass them over here and we'll collect those. But as we head towards those questions from the audience, I must admit one of the things that comes to mind for me is the potential parallel of your work with the current opioid epidemic. So I was wondering if you could comment about that.
I'd be happy to. Can I hate to take those pictures down. So there's incomplete information about the risk-benefit of these drugs used long-term. We know a lot about the risk from observational studies, but randomized trials with outcomes tend to be short. Most of them are 6 weeks, sometimes 8 weeks. 3 months is a really long-term trial for opioids. So we don't know that they work well for chronic pain in the long term. We've had, thanks to the Sacklers and Purdue Pharmaceutical, we have the OxyContin generation of physicians who were trained to turn very quickly to long-acting opiates for non-cancer chronic pain. And this has created basically a public health disaster. There are lots of things we know don't work. Included in those, I mean, everybody wants to help. So, legislatures want to pass limits on prescriptions and all kinds of things. And everybody wants to help, but most things have not worked, including CME, continuing medical education. That's been almost totally useless. And one of the things that concerns me is you can see the purple line, the prescription overdoses from the opioids have leveled off. And so what has happened is the heroin and fentanyl have taken over, and now the total mortality is going up. And what's happening is people are getting thrown out of the clinical care system, and they go to the streets. The drugs are cheaper there, but they're also quite dangerous. And what we need to do, and what we're doing at Harborview, for instance, is trying to manage the risk-benefit of the pain management as a whole. So we have pharmacists who track the opioid prescriptions for all the prescribers. And they're always coming up to me, does this one need this? And we have an opioid review committee where we can go to with problems. Jim Krieger remembers these. We have now pain management specialists. We can send our patients to pain management specialists. We have addiction treatment. So we're trying to keep them in care and not say, oh, we can't prescribe opiates to you. Go out in the streets and get them. And I think what we need from, this is a problem that the pharmaceutical industry and the medical community created. The FDA can't do much here. I really don't think so. Maybe at most they could try to limit some promotion, but this is something that medical care systems need to address. That's a long answer, sorry.
No, that's okay. But somewhat related, I guess.
Make them easy, it's late, it's almost my bedtime.
Well, some of these, I'm wondering, given your experience and what would your recommendations be about the FDA? Because certainly there's limited money. Who's
Around
responsible?
opiates?
Opiates, or in general, I guess. Overall, how might the FDA position itself better to reduce risk?
I think they've adopted the Sentinel Initiative, and I think in general they have done a very good job. I might pick on some particular cases that they've done, but they have some very difficult problems. Do anesthetic agents cause brain damage to children, young kids? They don't know, and they're wrestling with this and trying to promote a clinical trial with that. Should they bring bovine heparin back? It was taken off the market because of BSE, the bovine spongiform encephalopathy. And now we only have heparin, the porcine heparin that's coming from China, and that's not entirely reliable. It's almost impossible to go into a hospital without getting some heparin. So we need a heparin supply. Do we need to bring back bovine heparin? So they have a lot of difficult questions.
All right, now come the challenging questions from the audience. So you started with a quiz for them, and so now they're going to be getting back at you with some questions. So one from the audience: are there lessons to be drawn concerning the use of surrogates? Specific characteristics of surrogates that would indicate that they could be a good surrogate for clinical outcomes, and how do we evaluate these?
How much time do you have? So that's very difficult. If you have a surrogate that is, a disease-causing agent, like, HIV, that's probably a pretty good surrogate. For blood pressure, we've got 4 different drugs. There are 4 different drug classes. They work by very different biologic mechanisms. And it's actually clear now that they don't reduce the risks of different types of events by the same amount. Some are a little better at stroke, some a little better at heart failure. And it's not possible to look at a single surrogate with one drug and generalize that to other drugs, possibly within the class. So other diuretics maybe from the one diuretic. But I'd be reluctant to generalize quickly to other classes that work by other mechanisms. So I think we need to be, so I'm from Indiana, it's close to Missouri.
All right, thank you. Regarding precision medicine, what do you think? Hype or hope?
Do I get one word?
Go with it however you'd like.
So, we have been trying to tailor treatments to patients from time immemorial. So there's not really a lot new here. We may have more biologic information. And basically you're looking at some sort of interaction. And so you need even larger sample sizes. You need to show that, in fact, if you've got some genetic variant or something, that knowing that actually improves health outcomes. So you've increased your sample size by a factor of 4 to satisfy me.
One more from the audience, and I'm going to need some help on this. Are they working on a drug to replace
Coumadin.
Coumadin, oh, okay.
Not to my knowledge. I mean, they have the new oral anticoagulants that look to be pretty good in many instances. So that may well replace Coumadin, not for valvular heart disease, but for DVT and for atrial fibrillation. So the answer is we have them already.
All right, very good. Any other questions from the audience? Okay, here we go.
And in the front, that was an excellent talk, Bruce. I had a question. In one of your slides, you showed that you demonstrated the engine for the faster approval for FDA was the HIV epidemic. But I would argue that that actually did result in meaningful improvement and shortening the death spiral that was happening in HIV. So would you see cases where the FDA would be aggressive in approving drugs for some sort of diseases?
Oh, yeah.
Versus other conditions that they need to be more careful?
Yes, and so there's no doubt that that helped. But the Congress required, Nana, you don't get to ask one. But Congress required that for all drugs. And so I think actually oncology is doing a good job. They're learning a lot about the new immune system checkpoint inhibitors and some of the genetic variants that may define cancer risk. And so I think oncology is doing a good job moving drugs through quickly. I do think we need the post-approval studies to show that they are actually doing what we hope they do. So I'm not against rapid approvals, but the situation where you had rapid approvals and for everything and no post-market evaluation was very dangerous. And it led to things like Cerivastatin and Vioxx.
Another question?
Thank you. So the changes you've described at FDA in terms of post-marketing follow-up, et cetera, kind of a positive evolutionary development, well justified by experience. I'm not a close follower of FDA, but in the last year plus, we have an administration that clearly has a policy, a high-level policy agenda that cuts against the idea of reliance on science, of emphasis on research, of the regulatory enterprise in general. Is this showing up in FDA? Do you see it coming?
So my condolences to the EPA. I'm not sure. I'm on the science board. The science board is supposed to meet 3 times a year, and we advise the commissioner. The purpose isn't to see if the FDA got the science right on a particular drug. We're trying to answer the question, is the FDA doing the right science? We have not been convened while Gottlieb has been in office. So we've missed the last 2 meetings. There is one scheduled in April. I don't know whether it will actually happen. There was a good piece about him in the New York Times about a week or 2 ago that was fairly positive as a Trump appointee. So I don't really know yet, but they haven't asked the science board to meet in 8 months.
Bruce, nice talk.
Thank you.
What can you say about the F of the FDA? What authority do they have? What can they do? What should they do?
You know, I really can't. My ignorance is broad and deep. We do have people on the science board who know foods. And the evaluation of devices is weak. So I just talked about a few drugs. So just a tiny slice of what the FDA does. That one's too long.
We have a 5-part questions. I'll give you a couple of the easy ones first. What is your PhD in?
English language and literature. My PhD dissertation was entitled History and Ideology in the History Plays About King John. And it purported to engage the transition between feudalism and capitalism in the Renaissance. King John, this is the birth of the history play. You guys laugh. These are the first history plays. So its great merit is that it's done.
I'm not going to have time to go through all of these questions. Is cow fat the binding agent in tablets?
Is what?
Cow fat, the binding agent in tablets.
That's above my pay grade.
All right, the final question. What does MPH stand for?
You got me.
Okay, fair enough.
Well, I think it's my opportunity now to thank you again, Bruce, for taking the time this evening and for everybody coming out. I want to appreciate Bruce and his contributions to public health and to science and to drug safety. And I'll just tell you all to have a safe drive home, or however you're getting home, be safe and travel well. Thank you.
Thank you all.